Very much, and it's been a great series of seminars running over the last few months, and I'm delighted to have the opportunity to share some of our work with you this afternoon. So the session this afternoon focused on graphs, networks and molecules. Most of chemistry relates to molecules, so that's quite a good wide-ranging remit. I thought I'd take the opportunity first to tell you about some work on pharmaceutical lead discovery as an active search in a space of labeled graphs, and I'll tell you a bit about what is a data-driven adaptive Markov chain approach searching for antagonists of an alpha V integrin. I'll tell you a little bit about the medicinal motivation there. Then we will have a little bit of a shift of gears, and in the midst of that, we'll have a little bit of a pedagogical break. I thought I'd talk a little bit more in detail about the Weisweiler Lehman graph kernel, as graphs was part of the remit for this afternoon, and then wrap up with a topic, some recent work that follows on quite nicely from what Julie was talking about just now in the context of vibrational rather than electronic spectroscopy, and a focus on large proteins in contrast to the sorts of systems Julia was considering. So let's dive straight in, and our interest in the medicinal chemistry side over a number of years has been in this disease called Idiopathic Pulmonary Fibrosis, and lung disease, sadly all too familiar at the moment, and indeed pulmonary fibrosis or scarring of lung tissue is likely to be one of the effects from those who have suffered from more serious symptoms of COVID. So idiopathic means, is a fancy medical word meaning we don't know where it comes from, a bit of a catch-all, and it's been a disease growing in prevalence over recent years, characterized by decreasing lung function, some other rather nasty looking symptoms, and most frequently presenting in middle-aged male smokers. As I said, idiopathic is a bit of a catch-all, so the causes are not completely well understood. They may be environmental, coming maybe from work-related environments. They may be due to microbial agents. There may also be some variants that have a genetic component. The prognosis is often not particularly good, and the survival rate can be lower than a number of common cancers. And it's really related to damage to the lungs or buildup of scar tissue, reducing the available surface area for absorption of oxygen. And the prevalence is about 50 per 100,000 and on the increase. Heading into the biochemistry of interest, the target proteins are these family of transmembrane receptor proteins. So we can see a schematic here taken from one of the many papers from the Springer group at Harvard Medical School who've done a lot of the structural biology. So we've got the transmembrane here, TM, and we've got this rather complicated biological machinery, a multi-domain system that adopts some very different conformational states, which in its entirety allows interactions between cells and interactions between cells and the extracellular matrix, involved in a variety of regulation in the cell cycle and ultimately in immune responses and scar formation, which is not limited to just the lung as an organ. One sees fibrosis in various organs with different members of the integrin family playing different roles in different organs. So a complicated multi-domain protein here looking like a pair of legs and labeled as such in this diagram, and we're going to be focused at the top end of the system shown here. A number of crystal structures are available. This is one also from the Springer group, and we can see a sort of zoom in here at the interface of the heterodimer, which consists of an alpha and a beta subunit. So these can be paired in a variety of different combinations. And in mammals, there are currently known 24 unique integrins, where the alpha and the beta partners vary. And we can see that it's actually at the interface where the natural substrate binds and where there's interest in developing antagonists to stop the cascade that ultimately leads to the buildup of scar tissue. We are going to be focusing primarily on alpha V, beta 3 and beta 6, although in collaboration with GlaxoSmithKline, there are data on several other alpha V variants as well. The natural substrate has what's often familiar in medicinal chemistry is a binding motif, an RGD binding motif. And at Nottingham, over a number of years, final year undergraduate chemists have been making a series of compounds based on this mimic here, where we've got an naphthyridine fragment mimicking the arginine interacting with a carboxylate from the protein here. We have an alkyl chain and an amide bond mimicking the glycine in the middle, and then at the other end of the molecule we've got a carboxylate that interacts with one of the metal centers in the protein. Schematically, then, our chemical structure looks like this, and the chemical space that was of particular interest in terms of synthesizing new derivatives was based on substitution on the aryl ring here. Stereochemistry here is important, and if one considers substitutions at each position on the ring, and if those substituents themselves can be one of these 5-membered heterocycles, and one can have substituents on those also, we end up with a chemical space on the order of 180,000, which is probably quite modest by many contemporary drug discovery efforts in the pharmaceutical sector, but it was big enough for us to do some proof of principle studies that I shall talk about. So the algorithm here is an active search in which we're not explicitly enumerating all possible compounds. The challenge is, can we efficiently navigate, in this case, a proof of principle size chemical space, but obviously we want to be able to extend that many orders of magnitude higher. Can we navigate this chemical space efficiently without doing an explicit enumeration? And we have on the right hand here side the component of the algorithm that really makes it quite fast is a probabilistic surrogate, which is a maximum entropy model, which does a very fast evaluation of possible molecules that might be considered based on the known information so far, and evaluated through a maximum entropy type of term. So one has a Markov chain type process that runs for tens of thousands of iterations very quickly, and out pops the candidate molecule, which then goes to what's called an oracle. So this could be experimental data potentially if this were done quickly enough. In our case, it's molecular docking that one can substitute obviously a different or more favored in silico model of one's choice. As it turns out, the docking was relatively challenging here and was actually quite a good test for how efficiently one could run this cycle. So one gets information back from the oracle, basically in the form of a hit or not a hit based on a threshold in the docking score. The model is updated with the new information, so you have a labeled paramolecule in its label as a hit or a miss, and that's added to the training sample then, which updates this underlying distribution. And then one goes around this generating candidate molecules in batches, and we can see in general terms on this slide the performance of the approach. So we can run the algorithm for a number of rounds. Typically 50 seems to be appropriate. A critical aspect is how one generates the compounds, and one can see then that compared to a baseline of random sampling, the enrichment steadily increases. So we run multiple batches at each round, and the antagonist or the potential antagonist molecules I showed you had this quite long chain in the middle, and the active site is such that both ends of the inhibitor need to interact with form key interactions in the active site, leading to quite an extended structure for the molecule. So one has to generate a large number of conformations. Typically we generate on the order of 10,000 conformations of the small molecule. So the docking is actually at the more challenging end of the spectrum in terms of what one might come across in a med chem sort of setting. So substantial acceleration. We basically have docked here then 500 compounds out of a possible 185,000. So that's the sort of sampling of the entirety of the chemical space. So we see compounds that beat the docking threshold is one thing, and then the way the algorithm's set up, one can sample more than once candidate molecules that perform well according to the algorithm. So one can find frequently sampled compounds, and this is one example here with the cyanide derivative on the ring, the chelate interaction between the carboxylate and the metal, the bidentate hydrogen bond with aspartic acid 218, and the right stereochemistry, or the stereochemistry is consistently observed experimentally, was also consistently observed in our docking here, and the molecules that were picked out in the active search. So a more negative docking score, and this is OpenEye Fred for those familiar with docking algorithms. More negative means better, and S enantiomers were always consistently better than the R counterparts. So from the synthetic work that had been ongoing at Nottingham for a while, we were able to calibrate the performance of the approach. So when we start the active search, there are no data. So there's no initial training set, if you will, and the chemical space is defined simply by the parent molecule and the derivatives that we allow the algorithm to make. So there's a sort of implicit definition of chemical space by the points of substitution and the sorts of chemical moieties that one grows off the substitution or attachment sites. So no information goes in at the beginning, and the only information that is provided to the algorithm during its operation is the docking score that comes back. So from a few years ago, there were about 30 compounds that the undergraduate chemists at Nottingham had made, and GSK scientists had assayed. And we could see from our search that we often found a pattern of 3,4 substitution, which matched with what was seen experimentally. Remember, on that parent compound, all 5 points on the ring are allowed. Meta position frequently substituted. Out of the 30 possible compounds that the chemists had made, 20 were found by the algorithm. So that's with only 500 docking evaluations out of a possible space of 185,000. And many of the proposed compounds conformed both with published and unpublished work. Now, in the table, we've got some of the compounds in 4 different categories. The parent compound with just the hydrogen atom has a PIC50 that's below 6, so that means worse than micromolar. PIC50 is minus log 10 as the inhibitory concentration there, so you want a number bigger than 6 to have something that would be promising in terms of potency. So that's the parent down there. These compounds here were all found. I showed you the picture of the cyanide derivative. So the docking score is bigger than the threshold that we used, which is a sort of empirically determined threshold. And we can see these compounds found multiple times in the search, reinforcing the suggestion from the algorithm that these would be active compounds. There were some compounds that were not found. A couple were not found simply because they were not in the chemical space defined by how fragments were grown onto the attachment points. So this compound 39 comes back on itself to form a second ring, and that was not in the generation of possible compounds. So that's completely understood why we didn't find it. Then there were a few compounds which did not quite meet the docking threshold, so as under the minus 11.75, but interestingly found multiple times in some cases by the active search. So generally we were fairly encouraged by this. Here is another compound with one of these 5-membered heterocycles on, which has been of interest to the Nottingham synthetic chemists, forming the interaction with the metal and the aspartic acid 218 and some hydrogen bond in the middle. Whilst the work was ongoing, it was a live drug discovery project at GSK, so they've been quite active in publishing papers and patents. So the GSK compound described in these publications has this different linker here, but the same sort of endpoint in terms of the RR ring and the heterocycle, and we can see an interesting overlay where there are some commonalities and some differences in the specificity determining loop, looking a little bit different there. Obviously, this would probably require X-ray crystallography on on the Nottingham compound to dig into that a little bit further. But certainly a lot of interest generated by the active search and the molecules coming out of that. So multiple active inhibitors were found corresponding to published and unpublished research. I didn't talk too much about the algorithmic detail, but was developed by collaborators in computer science. There's a sound basis in machine learning, proposes structures from an implicitly defined space or so-called intentional space. There's a guarantee on the convergence, and provoked some significant interest that is still being pursued as we speak. This work was then developed by thinking about receptor flexibility. Hopefully you can see the receptor jiggling around there from some molecular dynamic simulation, so fully atomistic. The motivation here was not just to doctor a single receptor structure, but to a set of representative structures from the simulation. And that work is ongoing at the moment, possibly not yielding anything particularly more encouraging than what we already had from the static structure. In a different direction, we have looked at full free energy perturbation calculations based on extending these simulations into that thermodynamic framework, and that work is described in this recent paper where Ellen, a PhD student in the group, is the lead researcher on that. The active search and docking was done by Steve in the group, a PhD student just writing up his thesis now, and Simon was the colleague at GSK who worked very closely with us on this, alongside Tom McAnally, the Nottingham colleague who oversaw the synthetic chemistry. Dina and Thomas and Rahman did all the initial work on the algorithmic development for the active search, which is described in this 2018 paper, and some of the more recent synthetic chemistry described in this paper last year. And the chemistry was done in our GSK carbon neutral laboratory on the Jubilee campus at Nottingham. And it's really a unique building in the world in terms of prototyping carbon neutral facility for doing synthetic chemistry for pharmaceutical drug discovery in a carbon neutral fashion. So there's a big interest in Nottingham in sustainable chemistry, and I'll come back to that right at the end of the talk. So I'm going to shift gears a little bit, and in the middle here, just take a few minutes to talk about the Weisweiler-Leman graph kernel in a bit more detail, possibly more detail than you might wish, but we won't dwell on this too long. But I thought it might be nice to give a bit more of a flavour of some of the guts of the Weisweiler-Leman graph kernel, which has become quite popular in many machine learning applications at the moment. So our probabilistic surrogate that makes the active search fast before we do an oracle evaluation is based on this conditional probability featuring these exponential functions. And within, this conditional probability is basically if we're given a molecule, what's the likelihood that it will be active. So we've basically got a binary property space where we turn that docking score into more or less a yes, no, hit or not hit So we're interested in evaluating this. And within these exponential functions, then we've got the Weiss-Feiler-Lehmann graph kernel for our molecules. And I thought I'd just discuss the detail of the graph kernel in some detail. So the graph kernel, and this lets us use kernel-based methods where molecules are described as graphs, is rooted in the Weiss-Feiler-Lehmann test for isomorphism of two graphs. And the key idea is once taking the labeled nodes, okay, so a molecule is a set of atoms or nodes and a set of bonds or connections or edges in the graph. So we augment the node labels by a sorted set of node labels of neighboring nodes, and then compress these augmented labels into a new set of short labels. So we'll go through that in a little bit of detail based on what's published in the literature. Weiss-Feiler, interestingly and rather horrifically, has a backstory which is quite shocking, which I thought might be of interest to share. He was a Russian mathematician who emigrated to the US, and in 1985 disappeared in Chile on a hiking trip, and foul play, shall we say, was believed to have taken place, but was never proven. But there's actually a short film that everyone can see on Vimeo called The Colony, based on his disappearance in 1985, and the facts of the matter that sort of were dramatized in the film are given in this this website here. So I was looking for a little bit of biographical detail and was somewhat shocked to stumble on this anyway. So the algorithm is described in detail in this JMLR paper from about 10 years ago now by this group here, and all these next few slides are taken directly from a key figure in their paper. So we've got our molecule that is, or our two molecules represented by the graphs here, where the different atoms are labeled differently, 1 to 5, and we can see the different connectivity. We are then going to assign a multi-set label by looking at how each atom is connected to what other atoms. So atom 1 is connected to atom 4, and we've got another atom 1 here also connected to an atom 4. So these nodes are now labeled as 1, 4. Atom 4 is connected to 1, 1, 3, and 5. So the label here on the node is now 4, 1, 1, 3, 5, and so on. So we assign the multi-set label, and I'm only going to show one iteration, but this could be done for second nearest neighbors and so on. So we then sort each multi-set. So all our labels now are sorted, and we compress them. So we just give each of those a number, and then we relabel the graphs with our multi-set compressed labels to give the subtree pattern. So now the labels on the nodes are really reflecting the connectivity rather than the initial atom type. So before, where these two atom 4s were the same, they're now labeled differently because the atom 4 is connected differently in the two graphs. We then write out our, or representation in terms of the counts of the original node labels and the counts of the compressed node labels. So we get two atoms of type 1 here. So we had a 2 on this count here, and so on and so forth. And then one constructs the kernel by forming the inner product. So we're just going to go through our two vectors here and multiply each corresponding element with itself. So we've got 2 times 1 is 2, plus 1 times 2, another 2, so on. In some cases we have a 0, so those terms drop out in the inner product. And if we add up 3 lots of 2 is 6, and 5 lots of 1 makes 11. So that gives us our subtree kernel here. So just a brief interlude exposing some of the guts of the Weisweiler-Riemann graph kernel for you. So I thought in the final few minutes to share with you some of our recent work on predicting protein infrared spectroscopy, and I think it follows on quite nicely from the very clear and nice work that Julia described in the first talk in the session. So we've been interested in predicting protein spectroscopy from structure for many years now, and this is a collaboration with Shao Mukamal at UCI and his postdoc at the time, Jun Jiang, who's now a full professor at University of Science and Technology, China in Hefei. So the motivation is connecting to how one might experimentally determine, measure, observe protein folding, crystallography, and NMR, and obviously providing lots of detailed atomistic information, increasingly cryo-electron microscopy contributing in that space also. And in this list of biophysical methods, optical spectroscopy would be a rather poor cousin in terms of spatial resolution, but of course there's a lot of interest in the time resolution that one can access using optical spectroscopy, and we've been interested in this area, particularly with a focus on electronic circular dichroism, which I haven't got time to talk about, but Francois was a student working both at Soleil and with us in Nottingham on both fast time resolved measurements with a super fast camera from Shimadzu, and on some simulations and calculations with us at Nottingham. But we are going to be focusing on infrared vibrational spectroscopy. There are a number of approaches to computing biospectroscopy. One could do full ab initio MD and analyze the trajectories to transform the appropriate correlation function there, and that's certainly tractable on small molecules. One might look at hybrid QMMM processes, especially if there's an active site region, or in this case it was a fluorescent molecule we were interested in, where there's clearly a specific region to be treated quantum mechanically, and the rest of the environment could be treated at a low level of theory. But when one comes to protein spectroscopy, such as the amide 1 band in the infrared, this is really arising from more or less equal contributions from chromophores across the protein. So one can't sort of just say this chromophore is important and the rest aren't. They all need to be treated on a similar footing, and this is a so-called exciton approach, which we've applied widely to many proteins, including some recently of interest in the SARS-CoV-2 proteins there. So calculation of the spectroscopy of a protein involves, in this exciton framework, involves setting up a a exciton Hamiltonian, which with terms that really should have some resonance with what Julia was talking about earlier. We've got some excitation energies. So each of the individual chromophores will have an excitation energy or a vibrational frequency associated with them, and this will vary depending on the local environment. And some coupling terms, analogous, but in the to what Julia was talking about in the electronic regime, but here we're in the vibrational regime, and also some transition dipole moments. And I have an eye on the time, so I will wrap this up shortly. So much like Julia presented, we have a multi-layer backpropagating perceptron with a large, with in this case 3 hidden layers, and I think 30, 60, 428 units in the hidden layers, and we generate a training sample from a ab initio MD simulation of 1 peptide unit in explicit water. We excise configurations from that, do the DFT calculation of the frequency, the vibrational properties that we want then the neural network to reproduce. So we have on the order of about 10,000 training points based on uniform and fairly sparse sampling of a longish trajectory. And then for the nearest neighbor coupling terms, we have a different molecules, this covalent dimer sampled again over several thousand configurations. There was a question earlier about the invariant representation of the molecule, and I think this is very much the same sort of strategy taken here, the so-called Coulomb matrix, related to the charges on the nuclei and their separations. This factor of this power of 2.4 for the diagonal terms come out of the literature, and I think out of some of the work that Julia was referring to. And then we've got basically the product of the nuclear charges over their separation, and obviously those separations vary as the geometry varies from the simulation. So straight on to why we might want to do this, and this is really then looking at longer timescale simulations of protein folding. So this might be on the upper row, we're looking at ubiquitin, a workhorse exemplar of protein folding, where one can then look at the denaturation. One can see the denaturation curve here, and the experimental data in black, nicely echoed by the computational data then. So these are simulations, atomistic simulations, and explicit solvent at the different temperatures. And this gives us some reassurance then that our empirical force fields for the conformational dynamics are reproducing structures that agree with the experimental measurements. On the bottom side row here, we've got then simulation of the unfolding of the trip cage protein, another small exemplar of protein folding, where one's got a large number of ensembles corresponding to different stages of fully unfolded S1 through to fully folded S100. And obviously one would like to be able to connect the simulation data directly to time resolved experimental infrared measurements. Okay, so bit of a whistle stop tour through that, the second part there, but in summary, shared a little bit about what we've done in active search on a live drug discovery project, and talked about the Weiss Feiler-Lehmann graph kernel, and I've shown you what we've done recently in terms of using machine learning to model the relationship between protein conformation and spectra. I've given acknowledgements largely throughout the talk, and I just wanted to take the last 30 seconds to advertise a couple of things. One is a recent book in the Royal Society of Chemistry, Theoretical and Chemistry, Computational Chemistry book series that I edit. So this volume put together by Hugh Cartwright came out last year, Machine Learning in Chemistry. So lots of interest, the 20 plus chapters in there, covering a lot of recent work in the field, which I'm sure will be of interest to many. And finally, Sammy, right at the beginning, mentioned I've been lucky enough to have been awarded a Royal Academy of Engineering Chair in Emerging Technology. So this is going to focus on machine learning applied to various problems in sustainable chemistry, harking back to the GSK Carbon Neutral Laboratory. And there's opportunities there, which I'd be grateful if you would share with your networks. Currently looking to appoint postdoc and indeed PhD students. So with that, I've finished.